Bismillahirrahmanirrahim. Today we do the specimen paper for the new syllabus, which is for the 2022 syllabus, and we're going to start doing the MCQ paper, which of course is in June, but I want to do it with you all before that so that you can have a look at it and see where you will have to change your study technique so as to cover the new topics which have come in the new syllabus. The first thing to know has been increased to 1 hour and 15 minutes. Now, this has only been from the 2022 syllabus. Previously, these MCQs. So you have to realize the MCQs are going to be more difficult than the previous ones. So this is one thing which you must understand. This is 1 hour and 15 minutes. Now let's look at question number 1. Which definition of the magnification of a drawing of a leaf is correct? Now, of course, I've marked the one which is correct, the size of the drawing of the leaf divided by the actual size of the leaf. But the question is, why are these wrong? You see, the question is, it said magnification of a drawing of a leaf. So there is this leaf drawing on which it says x10. So this is the magnification. That means from the original leaf, this is 10 times bigger. So the actual size of the leaf multiplied by the magnification of the microscope? No, it said magnification of a drawing of a leaf. So this is wrong. Then the measured difference in size between the leaf and a drawing of the leaf. The measured difference in size. Say this original leaf was 1 centimeter, and now this is 10 centimeter. So the difference will be 9 centimeter. Well, that is not the magnification. So this one is wrong. Then the increase in size of the leaf when observed using a microscope. We see anything with a microscope was wrong. Why was it wrong? Because it said which definition of the magnification of a drawing of a leaf is correct. Well, basically, they're checking your English more than they're checking the biological concepts. Then coming on to question number 2, the electron micrograph shows parts of 2 cells. Now we just do the labeling first. W is a chloroplast and X is the cell wall and Y is the cell membrane. Always do that on the paper. Which labeled features identify these cells as eukaryotic? Now they're basically telling you to differentiate between eukaryotes and prokaryotes. You know, prokaryotes have cell wall and cell membrane. So the differentiating feature which makes this is a chloroplast. But you know why chloroplast? Because it's a membrane bound organelle. And eukaryotes have membrane bound organelles while the prokaryotes do not have membrane bound organelles. So that was the reason why the answer was C. Then it says plant cells are stained and viewed using a student's microscope. The light source is natural light, which could be clearly visible in 400 magnification. You realize that anything with less than the wavelength of light is 400 nanometers. And you can only see things which are larger than half the wavelength. So anything less than half the wavelength, which is anything less than 200 nanometers, well, you can't see it because it doesn't interfere with the light waves. So the grana and the cristae, these are all wrong, and ribosomes are of course only in nanometers. So the only answer was nucleoli, because cristae, it's not the mitochondria, it's the cristae of the mitochondria, means the inner foldings. This is the cristae of the mitochondria. Now that's a phospholipid bilayer, 7 nanometers. Similarly, the grana of the chloroplast is also thylakoid membranes. Grana wouldn't be that big. So this is why the answer to this was C. Now coming to question number 4, which lengths are equivalent to 1 micrometer? Well, you've got to know this, you've got to learn this. 1 centimeter is equal to 10 millimeter, 1 millimeter is equal to 1000 micrometer, and 1 micrometer is equal to 1000 nanometers. So 1 micrometer would be what? So this is why you've got to understand is that all these would be wrong if you could do all these calculations and do this. So the answer was only 0.001 millimeter, because 1 millimeter is equal to 1000 micrometer. So 1 micrometer will be 1000th of a millimeter, 0.001 millimeter. So this is correct. And the fact that 1 micrometer is 1000 nanometer, this was correct. So only 3 and 6 were correct, and that's why the answer was D. The rest are all wrong, and I'm sure you can do the calculations. Question 5. Some secretory cells synthesize and release glycoproteins. You know glycoproteins, glyco, G for glycogen, G for Golgi apparatus, proteins made on the fixed ribosomes. Because you remember, if anything has to be thrown out of the cell, it has to be working on this, an extracellular maybe protein. It has to be then made on the fixed ribosomes, because that's going to be packaged and is going to be sent to the Golgi, and then the Golgi vesicles are going to bud off, and then it's going to go to the cell membrane and exocytosis. So we all know this very clearly. Exocytosis would be the last thing. You don't have many choices in that being the last thing. There's only choices between B and D. Okay, what is the correct sequence of some of the events that occur in a secretory cell? Now, how do you start going about this question? The first thing is, it's mRNA binds to ribosome because that's where the protein is going to be made. So this is the ribosome, and here the protein is going to be made. So mRNA binds to ribosomes, right? And then synthesis of glycoproteins. Now they have sort of shortened it all up very and made it so this is going to bud off, this is going to be the secretory vesicle, and then this is going to go to the Golgi. The Golgi are the stack of these things. So this is finally going to go to the Golgi, then this is going to go and get attached here, and then this is going to enter the protein is going to enter this, and then the Golgi is going to add a glycogen part to it, and then it is again going to bud off. So it's going to break off, and this is going to go to the cell membrane, where there is going to be exocytosis of this glycoprotein, right? So, I mean, you got to understand is, if you know the subject, only then can you really understand it and understand how it is going to happen. So the first one was 3, then it's 4, synthesis of glycoproteins. They've shortened it, they haven't shown you. But this was the point which was the difficult point, because you see it said products accumulate. So that means now the Golgi has done its work and added the glycogen part. So the glycoprotein has been made. It said products accumulate in these secretory vesicles. Yes, secretory. This is the secretory vesicle. And then exocytosis. So please understand, read the question very carefully and understand which one would come first. So that's why it's 3, 4, 2, and then 1, which is finally exocytosis. Question 6. The scientist used a blender to break open a sample of animal cells and release the organelles. The extracted mixture was spun in a centrifuge at a low speed. The heaviest type of organelle sank to the bottom of the centrifuge to form a solid pellet 1. This is the solid pellet 1. And then this was poured into another test tube, this liquid above the pellet. The liquid above the pellet was poured into a clean centrifuge tube and spun at a high speed. Centrifuge spin the fluid at a high speed so that the solid particles settle out. The next heaviest organelle sank to the bottom to form pellet 2. So this was pellet 2 now. Pellet 2, pellet 1. Basically, it's the ones they're wanting you to find out whether you know the sizes of the organelles. The scientists repeated this procedure twice more to obtain pellet 3 and pellet 4, each containing a single type of organelle. That's again a question, single type of organelle. What is the function of the type of organelle extracted in pellet 3? Now, pellet 1 would be the nucleus. Pellet 2 would be the mitochondrion, pellet 3 would be the lysosomes, and pellet 4 would be the ribosomes. So you got to understand, this is that they're checking whether you know the sizes and which one is a larger and which one is smaller. So if they had asked you pellet 1, then the answer was this. You see, digestion of old cell organelles is lysosome, production of ATP by respiration is mitochondria, synthesis of mRNA takes place in the nucleus, and synthesis of proteins takes place on the ribosomes. So what is the function of pellet 3? They didn't ask you that. They asked what is the function of pellet 3. But unless you knew this, you couldn't have done this question if you didn't know this, what they were asking you, and how they were saying pellet 1, pellet 2, and pellet 3 and 4. What is the general formula for amylose? Now you know amylose is made up of what? Alpha glucose joined together. So what is it? C6H12O6 plus another C6H12O6. What exits it? Water. So minus the water from it, what do you get? C6H10O5 raised to the power n. Simple way of doing it. Question 8. Which statement describes how the molecular structure of starch is suited to its function? You see, starch is made of amylose and amylopectin. So you've got to know the structure of it. The fact that it is insoluble and does not affect the water potential of the cell, because starch you have to store for long times. So the store has to be number one, it has to be compact. Number two, it has to be insoluble. It should not affect the osmotic, the water potential, so that water doesn't move in by osmosis. Now, why are all these wrong? A, amylose has a branch structure, and amylopectin is coiled to give a compact molecule. Well, amylopectin is branched. Amylose is not branched. In the breakdown of amylose and amylopectin, many condensation reactions release stored energy. They didn't ask you that. They said how the structure is suited to its function. Basically, it's the structure of starch is a storage molecule. Why is it suited for its function? Then in the formation of amylose and amylopectin, many hydrolysis reactions occur. In the formation, well, we're not interested in the formation or that. We're interested in why is it very good for as a storage molecule. Here, please remember condensation reactions do not release the stored energy. It's only when the glucose is respired then release energy is released. A student who's not clear about this would make a mistake of picking this up. Now, coming to question number 9, cows and whales are mammals that produce milk to feed their babies. Newborn whales grow faster than newborn cows. So they've given you this information in the question that they grow faster. So newborn whales grow faster than newborn cows. Milk contains fatty acids with a range of chain lengths. The table shows a percentage of fatty acids of different chain lengths in cow milk and whale milk. Now, chain length 4 to 12, 14, 16, 18, more than 18. Which statement about the ratio of short fatty acids? So 4 to 16 is the short one. So I have done circle them in green. To long fatty acids, 18 or more carbon atoms in the milk of cows and whales is correct. So I've circled this to make you realize how this is correct. So you can see how there is a difference in it, that the short ones are less in the whale. So you have to really calculate this and then look at the answers. So the ratio in cow milk is higher because newborn cows need more energy. But then they had said that newborn whales grow faster in the question. Then the second, the ratio in cow milk is lower because newborn cows need less energy than newborn whales. Well, that's correct, but the ratio in cow milk is lower. It's not lower, it's higher. You can see this is more. So you had to look at these and then you had to really read these answers. Then the ratio in whale milk is higher because newborn whales need less energy. Again, that's wrong. That's in the question. They've given you that. So that is why the answer to this was D. The ratio in whale milk is lower because newborn whales need more energy than newborn cows. Fine. Coming to question number 10. The structure of phospholipid includes number one, polar phosphate heads, number two, hydrophobic fatty acid chains, number three, saturated fatty acid chains. Now, what are essential for the formation of a phospholipid bilayer in a cell membrane? Phospholipid bilayer is this. You see, whether they are saturated or unsaturated doesn't matter. It's the polar phosphate head and the hydrophobic fatty acid chains. The saturated may be saturated, may not be saturated. So 3 is not really actually needed. So what is essential for the formation of phospholipid bilayer? So that was only 1 and 2. Now coming to question number 11, the diagram shows the molecular structure of a peptide. Which molecule would result from the complete hydrolysis of the peptide? Now you can see there are how many the peptide bonds. This is the first amino acid, then this is the second amino acid, and then this is the third amino acid. Now the ones which are wrong is you can see how it said A, this CH2, there's no CH2 in that one, there are two. In B, there is not an OH here, which is missing, which I've done in green. And C was correct and D was wrong because this was a CH3. You see, you can compare this with this. This was correct. So CH3, there's no CH3 here. So you can see how these three were wrong and this one was correct. Question 12. The diagram shows a hemoglobin molecule, beta globin chain, You can see the alpha globin chain and the beta globin chain, alpha globin, beta globin. These have been underlined. The white ones are the alpha and the black ones are the beta globin. And then they've numbered some things 1, 2, 3, and 4. Now, 3 was definitely the alpha helix. But then you have to see what was 2 and 4. Well, 2 and 4 were the outside ones, and 4 was the inside one. So the 2 are the hydrophilic R groups, and 4 are the hydrophobic R groups. So you have to be very careful how you're going to be looking at this. They have 3 is the ones which are on the outside. Now, if you remember, a globular protein is something which is round. And what you have to have is you have to have the hydrophilic R groups on the outside and the hydrophobic R groups on the inside. So the hydrophobic ones are the ones which are on the inside, and the ones which are on the outside are the hydrophilic ones. So the hydrophilic ones would be these ones which are on the outside. Now, if you knew this only, then you could have done this question in which says D is the oxygen binding site, and 2 is the hydrophilic amino acids, and this is the alpha helix. Of course, I would have figured it out was the alpha helix. I would have definitely known that 3 was the alpha helix. So alpha helix, and the alpha helix was only in B and C. So I would have narrowed it down to these two and then thought about the question and then done the question. Now, question 13, a student investigated the effect of enzyme concentration on the rate of hydrolysis of protein in milk. When the enzyme and milk were mixed, the protein was hydrolyzed and the mixture changed from cloudy to clear. Now, you understand why cloudy to clear, because if the protein is converted to amino acids are soluble, so it becomes a clear solution. The student investigated 5 different enzyme concentrations and recorded the time taken for the mixture to become clear. Fine. So what are we measuring? The rate of disappearance of the substrate. What is an appropriate control for the investigation? Whenever we're doing enzymes, so we say, okay, control means comparison. So the enzyme must either be boiled and cooled, or it must be, instead of enzyme, we use a water solution. If still the reaction takes place, then of course we know it's not the enzyme, there is something else which is working. So what is an appropriate control? Carrying out an experiment where the enzyme solution is replaced with water. So that was the answer. But why were these wrong? Why was B wrong? Carrying out each experiment in a thermostatically controlled water bath at 35. This would be controlling the variables. Repeating each experiment 3 times, this is to make it more reliable, right? Using the same volume of enzyme solution for each of the 5 experiments. Yes, that is standardizing the variables. That is nothing to do with the control. That is to make sure that you have the same substrate and the same number of active sites and the same number of substrate molecules. So that is the volume and concentration is a standardization of the variable. Then coming on to question number 14, what determines the specificity of an enzyme? The covalent and other bonding between R groups of the polypeptide. Specificity of the enzyme means that this enzyme is specific. Why is it specific? Because it has the depression, the active site. Now, the active site is only possible because of the bonding and because of the 3D structure. So, the covalent and other bonding between R groups of the polypeptide. The optimum pH of the enzyme has nothing to do with the specificity. The covalent peptide bond between amino acids of the polypeptide, well, that could be, just maintaining the structure, the shape of the substrate molecule. No, that's all rubbish. So what we have to understand is that determine the specificity of an enzyme is the shape of the active site, which is the specificity of the enzyme. So the answer was C. Why was 3 wrong? Why? Because this is the covalent peptide bond between amino acids of the polypeptide. This is going to only ensure the primary structure. So the primary structure is the peptide bonds between the amino acids. So that is not going to be, that is not, the folding is done by the other bonds. This is, of course, the polypeptide chain has been made. So this would be in every protein. But the further folding, which results in an enzyme having this active site or an enzyme having this active site, is because of the bonding between the R groups of the polypeptide. So please understand why 3 is wrong. Now, coming to question number 15, the graph shows the effect of temperature on the rate at which the enzyme in a biological washing powder digests and removes the fruit juice stains. This is the rate of reaction. So now look at it how is it. The rate is increasing before T. The rate is decreasing after T. Which statement explains the shape of the graph at temperature higher than T? Now, what you're not going to read is explains the shape of the graph. The shape of the graph is the rate is decreasing. Bonds are broken between the R groups of the amino acids in the polypeptide chains of the enzyme. There are more collisions between the enzyme and the substrate. Now, that is not going to decrease the rate. So that was wrong. But would you be able to pick this up? Because you didn't read the question. Shape of the graph. The shape of the graph is rate is decreasing, but if there are more collisions, the rate should increase. So that is why this was wrong, and 1, 3, and 4 were correct. In question 16, which statement describes carrier proteins in cell surface membranes? Carrier proteins are found on the outer surface. No, they're found anywhere. They could be outer, inner, they could be spanning right through the membrane. Carrier proteins are involved in moving substance through the membranes by either active transport or passive transport. Carrier proteins allow the binding of ligand molecules which cause changes within the cells. Not all carrier proteins do that. It would be a receptor protein, couldn't be a carrier protein which acts as a binding for the ligand. Please remember, ligand is something new in the syllabus. Carrier proteins are involved in moving substance through the membranes by passive transport through water-filled pores. Oh, that was total rubbish. So you have to understand why carrier proteins. Carrier proteins are moving substances, could be by facilitated diffusion, could be by active transport. So it's not necessarily all active transport. Question 17. What happens to a typical bacterium when it is placed in surroundings that have a higher water potential than the water potential inside the cell? So this is a bacteria, and you have 1% solution outside which has a higher water potential, and inside is a 3% cytoplasm. So there is going to be a net movement of water into the bacterium. You have to understand, bacterium has a cell wall and a cell membrane. So this was wrong when it said the bacterium bursts because the cell wall has no structural function. The bacterium dies because water leaves the cell. No, water is going to enter the cell. Water is not going to leave the cell. So this was total rubbish. There is no change because cell wall is impermeable to water. No, cell wall is fully permeable to water. So this was wrong. So there is a net movement of water into the bacteria. More water enters, less goes out. There is ever not only one. There's always, you can't say there's no movement or there is movement one way. There's both ways, but the net movement is more because it said surrounded by a higher water potential than the water potential inside the cell. Then question 18. By which process do hydrogen carbonate ions leave the red blood cells? Well, they would naturally leave it by facilitated diffusion because it's an ion, and it diffuses out of the red blood cell. So they have special channel proteins for that. So facilitated diffusion, and because they are ions, they would not be able to pass the hydrophobic part of the cell membrane. So they would have to have a channel protein through which it is going to move out. Then question number 19. Question 19. In an experiment, fluorescent dyes were attached to proteins on the outer surface of cell membranes. Fluorescent dyes of one color were attached to proteins of a living human cell, and fluorescent dyes of a different color were attached to proteins of a living mouse cell. So let's look at maybe green was the dye color here, and red was the dye color here, were attached to proteins of a living mouse cell. The human cell and the mouse were then fused to form a hybridoma. So we would have this and we would have this. At first, the proteins attached to different fluorescent dyes remained separate, but after 40 minutes, the proteins were distributed randomly across the hybrid cell surface membrane. So we had something like this. At first, the proteins attached to different fluorescent dyes remained separate, but after 40 minutes, the proteins were distributed randomly across the hybrid cell surface membrane. What does this experiment show? Proteins move freely in the phospholipid bilayer. So you have to understand this were attached to proteins on the outer surface of the cell membrane. So they were randomly, because the proteins move randomly in the phospholipid bilayer. That is one of the syllabus points that you are supposed to learn, the fluid mosaic model. Then coming to question number 20, the diagram shows the mitotic cell cycle, during which phase is DNA replicated. Now, of course, you have to be very clear with this. If it is cytokinesis, if this is cytokinesis, then before that has to be mitosis, prophase, metaphase, anaphase, and telophase. This is mitosis. So C, this is A, B, and C are all interphase. A is G1, B is S phase of interphase, and this is where DNA replication takes place, and this is G2. So S phase of interphase, so that's why the answer was B. Now I conclude this video here, and I will continue the rest of them on another video, and thank you very much.